Hello, hi friends, good morning. Welcome to our channel, The Voice of Nurse. Today we are going to discuss a continuous topic from the previous video, microbiology. This is part 2. We already discussed part 1 in previous video. In part 1 we discussed related to the introduction of the microbiology definition, what is microorganisms, types of microorganisms, and what is infection, its transmission, immunity and types of immunity, and who is father of microbiology, who is father of antiseptic surgery and sterilization techniques introduced by. These all things we discussed in part 1. Along with part 1, these introduction plus 20 questions we discussed in previous video. Today class we are going to discuss continuous questions from the previous videos, 21 to 50 questions. 30 questions we are going to discuss in this video. This is part 2. Before today class discussion, I had to inform to all about MHSRB Telangana staff nurse recruitment. Recently 2 days back announced about Telangana state MHSRB staff nurse exam will be conducted in the online process, not in offline process. They will conduct exam in online process. Within 1 week we will get a exam date. Then for exam date and exam date release date, means after 1 week we will get exam date, then it will be takes 2 months, approximately 2 months time for the conducting exam. Just 2 days back we got a news, that's why I am informing to you all. So we are having MHSRB exam in online process, means computer exam. There is no offline process recently released. So be careful friends. Within 1 week we will get a exam date. Okay friends, be prepared friends. Today we are moving on to topic, the topic discussion, microbiology. Before moving on to topic, a small request friends, anybody watching first time my channel, please subscribe my channel, click on the bell icon for further notification, and share the video to your friends and share your opinion in comment section, and please give a like. Let's move on to topic. Today we are going to discuss a questions from the previous video from continuous topic of microbiology FAQs. 21 question, antibody is responsible for allergic reaction or response. So antibody is responsible for allergic response. We will see here options. Option A, IgA, immunoglobulin A. Option B, immunoglobulin E. Option C, immunoglobulin G. Option D, immunoglobulin M. So here, which antibody is responsible for allergic reaction means antibody is responsible for any allergic reaction is immunoglobulin E. If our body contains any type of allergies or immune system overreacts to the allergy by producing antibodies, it is called as immunoglobulin E. Means our body contains any type of allergy, our immune system overreacts allergies by producing antibodies is called as IgE. The antibody has a Y-shaped structure. Antibodies can be classified into 5 types. Those antibodies are immunoglobulin A, immunoglobulin M, immunoglobulin E, immunoglobulin G and immunoglobulin D. So these all are the types of antibodies. But antibody is responsible for allergic reaction. Any allergic reaction is immunoglobulin E. So here in these options, option B is the correct answer. Next we will move on to question number 22. Dental caries caused by. The dental caries caused by. We will see here options. Option A, streptococci. Option B, shigella. Option C, lactobacilli. Option D, staphylococcus. The dental caries develops when bacteria in the mouth metabolize sugar to produce acid that demineralized the hard tissue of the teeth. The dental caries develops when bacteria is in mouth metabolize sugar produce acid that demineralize the hard tissues to the teeth. Most important bacteria is causing dental caries is lactobacilli and streptococcus, but major cases due to the lactobacilli. So here in these options, the dental caries is caused by option C, lactobacilli is the correct answer. Next we will move on to question number 23. Endoscopes are sterilized by. The endoscopes are sterilized by. We will see here options. Option A, autoclave. Option B, 2% glutaraldehyde. Option C, formaldehyde. Option D, all of the above. Endoscopic instruments do not tolerate autoclaving, a new method of sterilizing by moist method. The new method of sterilizing by moist heat method has been used in the laboratory and shown to be an improvement. 2% glutaraldehyde is used for manual and automated disinfection. 10 minutes immersion is recommended for endoscopes before the session and between the patient's usage. So here the endoscopes are sterilized by using or by method of glutaraldehyde. So here in these options, option B, 2% glutaraldehyde is the correct answer. Next we will move on to question number 24. Sleep sickness caused by. What is the cause of sleep sickness? We will see here options. Option A, blood transfusion. Option B, testis fly. Option C, mosquito. Option D, person to person. The African trypanosomoniasis. The African trypanomoniasis is also known as sleeping sickness. It is caused by microscopic parasites of the species Trypanosoma brucei. It is transmitted by the testis fly or Gloxinia species through the infected parasite testis fly bites. Why it is called a sleep sickness? The sleep sickness, why it is called as sleep sickness, means because of symptoms can include say disturbed sleep in the pattern. That's why it is called as sleep sickness. So here in this option, sleep sickness is caused by option B, testis fly is the correct answer here. Next we will move on to question number 25. Through the autoclave method, what we will destroy. So while using the sterilization method, one of the autoclaving method is one of the type of sterilization method. What we will destroy through the autoclaving method? We will see here options. Option A, all microorganisms. Option B, yeast. Option C, pathogenic microorganisms. Option D, non-pathogenic microorganisms. Here, while we using the sterilization method, automatically we will destroy the kill harmful bacteria like bacteria, fungi or spores. So we have to destroy all microorganisms. So here in this option, option A is the correct answer. Through the autoclave method, we will destroy the microorganisms. Autoclaving is the most effective method of sterilizing the lab equipment, especially for liquid handling products to kill harmful bacteria, viruses, fungi and spores. So here in these options, option A, all microorganisms we will destroy through the autoclaving method. So option A is the correct answer here. Next we will move on to question number 26. Method of transmission of syphilis is. What is the method of transmission of syphilis? Method or mode of transmission of syphilis. We will see here options. Option A, contact. Option B, congenital. Option C, A and B. Option D, none of the above. Syphilis spreads from person to person by direct contact with a syphilitic sore known as canor. It can occur around the penis, vagina, rectum, anus, lips and or mouth. Syphilis can spread during the vaginal and anal sex. It can spread to the fetus of person who has syphilis during the pregnancy. So here the mode of transmission or method of transmission will be contact through the sexual contact, and congenital also we will get, means mother to fetus. So here option A and B is the correct answer. Congenital and contact to the sexual and congenital to be mother to fetus, but most cases is congenital to the mother to babies. 60 to 80% of cases probably is mother to fetus, means congenital is more transmission in syphilis, and contact through the sexual will be 30% cases we are notifying. So here method of transmission of syphilis is option C, A and B is the correct answer here. Next we will move on to question number 27. Principles involved in autoclave. The principles involved in autoclave. What is the principles by using the autoclave? Means what we are following a principle. We will see now first options. Option A dry heat, option B moist heat, option C steam under pressure, option D B and C. The autoclave method will work on the principle of moist heat sterilization. The high pressure inside the chamber increases the boiling point of water for the sterilization of equipments. The higher pressure also ensures the rapid penetration of heat into the deeper parts of equipments. The basic principle is involved in autoclaving method is moist heat. So here in these options, the principles involved in autoclave method is option B, moist heat is the correct answer. Next we will move on to question number 28. Separation of a single colony is called as. The separation of a single colony is called as from the options. We will see here options first. Option A separation, option B pure culture, option C isolation, option D A and C. A separation of a single colony is called as isolation. Isolation refers to separation of single strain from a mixed population. So this help in separation of single bacterial colony. Why we have to do a separation means isolation precautions. We have to follow isolation precautions. Preventing transmission of infectious agents in healthcare settings. What is preventive transmission means? For example, through the contact or droplet, it will be transmitted to the one person to another person, means they will be most of the cases respiratory communicable diseases we have to keep in isolation. For example, for the preventing transmission measures, the conditions like tuberculosis, COVID-19, dengue and measles, viral hepatitis, all airborne or droplet infection comes under the mode of transmissions. So here in these options, option C isolation is the correct answer. Separation of colony or separation of single colony is called as isolation. Next we will move on to question number 29. Presence of virus in the bloodstream is known as. It means the virus is present in the bloodstream. So the word in the medical term, what we will call. We will see here options. Option A bacteremia, option B viremia, option C pyemia, option D none of the above. So what is bacteremia? Bacteremia means bacteria in the blood. So presence of bacteria is present in the bloodstream is called as bacteremia. Next option B, what is viremia? Viremia means the presence of virus in the blood. So here in this option only correct answer, presence of virus in the bloodstream is called as viremia. Next option C, what is pyemia? Blood poisoning. Pyemia or septicemia. Pyemia is also called as septicemia. Pus forming bacteria released from an abscess is called as pyemia. So here in this option B viremia is the correct answer. We will see in detail. Viremia, the presence of viruses in the blood. Uncontrolled virus proliferation leads to chronic viremia. Viremia is caused by a virus. Many different types of viruses can cause viremia. A virus attaches one cell, releases its DNA or RNA, takes control of cells and forces it to replicate the virus. For example, for the viremia is measles, but most common cause blood infection leads to most cases is bacterial infection. For example, for the bacterial infection is sepsis. But in this options, presence of virus in the blood is called as viremia. So here in this option B is the correct answer. Next we will move on to question number 30. The 30th question is example of killed vaccines. What is the example of killed vaccine? We will see here options. Option A, pertussis. Option B, rabies. Option C, hepatitis. Option D, all of the above. In this option, what is example for the killed vaccine? The killed vaccines are made from a protein or other small pieces taken from a viruses or bacteria. The killed vaccine are flu or polio or rabies, DPT, diphtheria, pertussis and tetanus, and poliomyelitis, hepatitis A or hepatitis B, Haemophilus, influenza type B. These all are the killed vaccines. Most common killed vaccine is whooping cough, pertussis, but rabies and hepatitis A also comes under the killed vaccines. So here in these options, the example of killed vaccine is all of the above is the correct answer. Pertussis, rabies and hepatitis. These all are the killed vaccines. Next we will move on to question number 31. 31st question is name of the oral polio vaccines. What is the name of the oral polio vaccines? We will see here options. Option A, BCG. Option B, TAB, option C, Sabin, option D, Salk. What is oral polio vaccine means? Oral polio vaccine, other name or in the form of oral polio vaccine will get in the form of which of the options? Here in the 1962 SARS vaccine was replaced by the Sabin vaccine. So oral polio vaccine or Sabin vaccine named for its inventor Albert Sabin. That's why the oral polio vaccine is called as Sabin. The oral polio vaccine is invented by the Albert Sabin. Intrapolio vaccines based on killed or inactivated poliovirus serotypes 1, 2, 3 was the first vaccine to break the sausage of polio epidemics in the year of 1950s. But the name of the oral polio vaccines in 1962 SARS vaccine, but after was replaced by the Sabin vaccine because that vaccine was named for inventor Albert Sabin. So here in this option name of the oral polio vaccine is option C. Sabin is the correct answer. Next we will move on to question number 32. Viruses require for growth. What are the requirement for the virus growth? We will see here options. Option A, plants. Option B, bacteria. Option C, protozoa. Option D, living cells. Viruses need to be inside a living cell to live and grow. Viruses are like photocopiers. They can make lots of copies of themselves. Once they are inside a host, the viruses will make lots of copies. Viruses are tiny infectious agents that rely on living cells to multiply. If the grow of virus like photocopiers means the lots of copies will take themselves means inside of the entering of the inside of a host the virus will grow in the living cells to multiply. So the viruses require for growth is living cells. So here in these options option D is the correct answer. Viruses requires for the growth is living cells. So here option D is the correct answer. Next we will move on to question number 33. Site for Montex test. What is site of the Montex test? Montex test we will go doing for the tuberculosis test, nothing but Mycobacterium tuberculosis. So here we will see options first. Option A, posterior left forearm. Option B, deltoid region. Option C, anterior left forearm. Option D, none of the above. The Montex test is also called as tuberculin test. The test performed by injecting 0.1 ml of tuberculin purified protein derivatives, also called as PPD. Purified protein derivatives, PPD, into the inner surface of forearm. The injection should be made with a tuberculin syringe with the needle level facing upward. The injection should be placed on the palm side up surface of the forearm, about 2 to 4 inches below the elbow. So, the site for Montex test is anterior left arm, left forearm. So here in these options, option C is the correct answer. Left forearm, anterior side of the left forearm is the correct answer. Next we will move on to question number 34. Which organism responsible for filaria? Which organism is responsible for the filaria? We will see here options. Option A, Anopheles microorganisms. Option B, Escherichia bancrofti. Option C, hookworms. Option D, all of the above. A tropical disease caused by the presence of filarial worms, especially lymph vessels. The causative agent for the lymph filariasis includes the mosquito-borne filarial nematodes, Escherichia bancrofti, Brugia malayi, and Brugia timori, and estimated 90% of lymphatic filariasis cases are caused by the Escherichia bancrofti. The filariasis is a rare tropical disorder caused by the roundworm parasites. Escherichia bancrofti or Brugia malayi is a responsible organism for the filaria. So here in these options, option B is the correct answer. Escherichia bancrofti is responsible for the filaria. Next we will move on to question number 35. Which condition is associated with Dick test? So here in these options, what is the condition is associated with Dick test? We will see here options first. Hansen's disease. Option B, typhoid. Option C, scarlet fever. Option D, tetanus. Dick test method of determining susceptibility to scarlet fever by injection into the skin of 0.1 cubic centimeter of scarlet fever to toxins. A reddening of the skin in the area over 10 mm or 0.4 inches in diameter within about 24 hours indicates lack of immunity to the disease. Scarlet fever or scarlatina, a bacterial illness that develops in some people who have strep throat and common in children, means those who are having developed, some people will get a strep throat. The sclerotic fever is common in children from age of 5 to 15 years of age group will develop a sclerotic fever most commonly. So here the condition is associated with Dick test is option C. Scarlet fever is the correct answer. Scarlet fever. So here option C is the correct answer. Next we will move on to question number 36. What is the generation time of Escherichia coli is? What is the generation time of E. coli? We will see here options. Option A 20 minutes, option B 20 days, option C 20 hours, option D 24 hours. Escherichia coli or E. coli is gram-negative facultative anaerobic bacteria. It is in rod-shaped, commonly found in the lower intestine of warm-blooded organisms. Generation times were calculated from growth rates using equation. Literature states that a generation time of Escherichia coli about to the 20 minutes. So here the generation time of Escherichia coli is 20 minutes is the correct answer. In these options, option A is the correct answer, 20 minutes. Next we will move on to question number 37. Oxidase positive bacteria are from the following. We will see here options. Option A, Vibrio. Option B, Pseudomonas. Option C, Neisseria. Option D, all of the above. Microorganisms are oxidase positive when the color changes to dark purple within 5 to 10 seconds. Microorganisms oxidase negative if the color does not change or it may takes longer than 2 minutes. Usually the family Enterobacteriococci gives a negative results, whereas Pseudomonas and Neisseria, Aeromonas, sap and Vibrio sap and Pseudomonas. These all are the condition oxidase will give a positive results. So here in this option oxidase positive bacteria will be Vibrio, Pseudomonas, Neisseria. These all are the options is the correct answer, means option D all of the above is the correct answer. A, B, C is the comes under the oxidase positive bacteria. Next we will move on to question number 38. Pathogenic organisms are option A important to industry, option B used in pasteurization process, option C harmful to human, option D none of the above. Pathogens are organisms that can cause disease. The different types of pathogens and the severity of the disease that the cause severity infections. Pathogens cause the illness to their host through the a variety of ways. So here the pathogen organisms will affect harmful to the human. So here in this option, option C is the correct answer, pathogenic organs harmful to the humans. So option C is the correct answer. Next we will move on to question number 39. Which of the following is a vector-borne disease? From the following is vector-borne disease. Which one is vector-borne disease? We will see here options. Option A diphtheria, option B rubella, option C dengue, option D tetanus. Disease that result from a infection transmitted to humans and others is called as vector-borne disease. Vector-borne disease means disease that result from an infection transmitted to human and others is called as vector-borne disease. The disease can be result from the infection transmitted to human and other animals by blood feeding arthropods such as mosquitoes, ticks and fleas. Example of vector-borne diseases includes dengue fever, West Nile virus, Lyme disease and malaria. The vector-borne diseases that put one of the every two people in at risk. So here vector-borne disease in the following options, vector-borne disease is dengue fever. So here dengue is the correct answer, option C. Next we will move on to question number 40. Which of the following not sterilized by? Sorry friends, here typing mistake is there. Which of the following not sterilized by autoclave? So here after sterilized by you have to mention autoclave. Which of the following not sterilized by autoclave? We will see here first options. Option A suction catheter, option B NG tube, option C Ralph's tube, option D scissor. Sharp instruments are not sterilized by autoclave. It is usually takes around 20 to 30 minutes duration, but for sharp instruments boiling should not be used because less reliable may affect the edges of shafts by making them blunt. Some surgical tools that are cleaned using hydrogen peroxide or the cutting and desiccated instruments like scalpels, scissors and saws, we can use this method. So here which of the following Not sterilized by autoclave means sharp instruments not be sterilized by the autoclave. Here in this option, sharp objective means scissor is there. Option D is the correct answer. So we should not sterilize the scissor through the autoclave method. Option C D is the correct answer. Next we will move on to question number 41. Mycobacterium tuberculosis is also known as. We will see first options here. Option A, magical bacilli. Option B, Hansen's bacilli. Option C, Cox disease. Option D, none of the above. Tuberculosis is caused by a bacterium called as Mycobacterium tuberculosis. The bacteria usually attack the lungs, but tuberculosis bacteria, Mycobacterium tuberculosis bacteria, can attack any part of the body such as the kidney, spine and brain. Tuberculosis is curable and preventable in the short period of time. So here Mycobacterium tuberculosis is also known as Cox here Cox bacilli or Cox disease. Tuberculosis is a spread from person to person through the air. So tuberculosis is also called as Mycobacterium tuberculosis and Cox disease. So here in this option, option C, Cox disease is the correct answer. Next we will move on to question number 42. All bacteria that inhibits the human body are. The bacteria will inhibit the human body. We will see first options. Option A, autotrophs. Option B, heterotrophs. Option C, phototrophs. Option D, chemilithotrophs. We will see one by one. What is autotrophs? An autotrophs is an organism that can produce its own food using light, water, carbon dioxide or other chemicals. So that is not inhibit the human body. Next we will see option B, heterotrophs. Heterotrophs. A heterotrophs is an organism that eats other plants or animals for energy and nutrients. Heterotrophs is Greek word hetero means other, trophs means nourishment. So heterotrophs is the correct answer. The bacteria inhibit the human body is heterotrophs. So here option B is the correct answer. Next we will see phototrophs. Phototrophs, an organism that uses energy from sunlight to synthesize organic components for nutrition, organism or protitis and bacteria. So this is not here. Next option D. Option D, chemilithotrophs means rock eaters and used to designated organisms generates energy by the oxidation of inorganic molecule for energy. So here a heterotroph is an organism that cannot produce its own food. So the bacteria that inhibit the human body is heterotrophs. Option B is the correct answer here. Next we will move on to question number 43. What is the best diagnostic method for acute hepatitis B infection? What is the best diagnostic method for the identification or diagnostic method for the acute hepatitis B infection? First we will see the options. Option A, HBeAg. Option B, HBcAg. Option C, HBsAg. Option D, all of the above. Hepatitis B surface antigen HBsAg is the hallmark of hepatitis B virus infections, and it is the first serological marker to appear in acute hepatitis B, and persistence of HBsAg for more than 6 months suggests chronic hepatitis B virus infection. A simple blood test can also determine if you are immune to the condition. Liver ultrasound, a special ultrasound called as transient elastography, can show the amount of liver damage. How much liver damage is happened we can identify through the liver ultrasound. A special ultrasound is called as transient elastography. But here in this diagnostic method for acute hepatitis B infection is option C, HBsAg. HBsAg. So here in this option C is the correct answer. Next we will move on to Question number 44. In DNA structure, adenine is linked with. In DNA structure, the adenine is linked with. We will see here options. Option A, uracil. Option B, cytosine. Option C, thiamine. Option D, none of the above. This is not thiamine, this is thymine. Okay friends, in DNA structure, adenine is linked with. We will see a double-stranded DNA molecule, adenine based on the stranded pair with the thymine bases on the opposite strand. Adenine is the one of the two purine nucleobases used in forming nucleotides of the nucleic acid. In DNA, adenine binds to thymine via two hydrogen bonds to consist in stabilizing the nucleic acid structures. In RNA, which is used for protein synthesis, adenine binds to uracil. So here in DNA is thymine. Option C, DNA structure adenine is linked with the thymine. Option C is the correct answer. Next we will move on to question number 45. Which of the following disease is caused by sugar cane? The cause for the sugar cane. We will see here options. Option A, asbestos. Option B, bagasosis. Option C, bicyanosis. Option D, cox. Here we will see one by one. Asbestos. Asbestos caused by breathing in asbestos fibers. People working in certain trades are more likely to have been exposed to the asbestos in the past. So here asbestosis is not a sugarcane. Next option B, bagasosis. Option B, bagasosis. Bagasosis is an airborne disease caused by inhalation of fibrous cane sugar residue is called as bagasosis. So here option B is the correct answer. From the following options, the disease caused by sugarcane is option B, bagasosis is the correct answer. Next we will see option C also. What is bicyanosis? Bicyanosis is a disease condition of the lungs. It is caused by breathing in cotton dust or dust from the other veg fibers. So this is bicyanosis. Cox means we know Cox disease is also called as Cox bacilli and Mycobacterium tuberculosis caused the tuberculosis. Cox is tuberculosis. So in these options, option B is the caused by bagasosis is caused by the sugar cane. Next we will move on to question number 46. If infection can be transmitted by the objects linked with clothing, toys, eating utensils, etc. is called as. We will see here options. Option A, indirect contact. Option B, direct contact. Option C, airborne. Option D, all of the above. A infectious disease, or if infectious agent from a reservoir to a host by suspending air particles, inanimate objects or inanimate intermediates is called as indirect contact. So here in this option, option A is the correct answer. A infection can be transmitted by the objects links with cloth, toys, eating utensils or any particles inanimated to the object is called as indirect contact. And also contaminated objects like food and drinking water, airborne transmission, animal to person. These all comes under the indirect contacts. Direct contact means through the direct contact with the infected person. If we are getting a infection or disease means it is called as direct contact. Airborne means through the droplets or through the air we will get droplets contact is called as airborne contact. So here option A is the correct answer. Indirect contact is the transmitted by the objects, links, clothing, toys, eating utensils. So here option A is the correct answer. Next we will move on to question number 47. Chain of spreading of malaria is. What is the chain of spreading malaria? We will see here options. Option A, man, mosquito man. Option B, bird, man to man. Option C, man to anthropod to man. Option D, man to pig and man. Malaria spreads when a mosquito becomes infected with the disease after biting an infected person, and the infected mosquito then bites a non-infected person. The malaria parasite enters the person's bloodstream and travels to the liver. So here the malaria spread through the mosquito become infected person to the uninfected person through the biting. So here man to anthropod to man. The chain of spreading malaria is man to anthropod and man. So here option C is the correct answer. Sometimes we will get in exams like man, mosquito, man. Like it is also chain of spreading malaria. So here option C is the correct answer. Next we will move on to question number 48. Frozen vaccine or frozen DPT vaccine should be, we will see here option A discarded, option B reuse after shaking, option C allowed to melt, option D A and C. DPT vaccine can be damaged by freezing, so we should discard frozen DPT vaccine. Before using medicine, by using the shake test, we have to assess the frozen DPT vaccine. Then smooth and cloudy, starting to clear, no sediment, we can use the vaccine. So after shaking the vial, then smooth cloudy, starting clear is there, no sediment, we can use the vaccine. And in case of not smooth, granular particles are present, thick sediment means do not use the vaccine because frozen present in the vial. So immediately we have to discard the vial. So here option A is the correct answer. Frozen DPT vaccine should be discarded. Reuse is not correct. So we have to discard. Option A is the correct answer here. Next we will move on to question number 49. Which of the following method is used to observe the motility of a microbe? So which of the method we can observe the motility of microbes? We will see here options. First option A wet mount technique, option B hanging drop method, option C A and B, option D none of the above. So here the method is used to observe the motility of microbe is nothing but hanging drop method. The hanging drop method is a special type of wet mount in which a drop of medium containing the organisms is placed on a microscope slide, often used in dark illumination to observe the motility of bacteria. Why? Why we have to see the motility of the microbe? The advantages like the wet mount, the hanging drop method preserves cell shape and arrangement. This is the advantage for the hanging drop method. So here in this option B hanging drop method, through the hanging drop method we can observe the motility of microbe. So here in this option B is the correct answer, hanging drop method. Next we will move on to question number 50. This is last question in this video. Find the mismatch from following infections. From the option, which one is mismatch? We will see here options. Option A rabies, dogs. Option B hydatid, cats. Option C plague, rats. Option D zoophilic dermatophytes to cats and dogs. Hydatid disease is a zoonotic disease caused by the intermediate stage of the dog tapeworm, that is Echinococcus granulosus. So it causes cyst formation in the internal tissue such as the liver, lungs and brain of the grazing animals and people. So hydatid disease, it is caused by the dog tapeworm and Echinococcus granulosus. So here option B is the mismatch here. Rabies is infection from the dogs, plague from the rats and zoophilic dermatophytes comes from the infection cats and dogs. So here hydatid is caused by Echinococcus granulosus. So this is the mismatch here, option B. This is the 50th question, is last question in this today's class. This is the part 2 related to the question number 31 to 50 questions related to the microbiology. Next, from the 51 question to, we will continue for the next class. Thank you so much for watching my video. Anybody watching first time my channel, please subscribe my channel, click on the bell icon for further notification, give a like and share the video. Thank you so much for everyone while watching the video. Give a like and thank you so much for supporting me. Stay tuned friends, keep watching.